Lovely. I think we'll get started. Hi everybody, and thank you all for coming to today's Journal Cluster of Neurosciences webinar titled Cognitive Impairment After Stroke, Current Research into Neuropathology, Evaluation and Management. My name is Meg, and I work for MDPI, helping to run their webinars. Before we get into the body of the webinar, I'll be giving a short introduction into MDPI, the Journal Cluster, and today's speakers. So a little bit about MDPI. A pioneer in scholarly open access publishing, MDPI has supported academic communities since 1996. Based in Basel, Switzerland, MDPI has the mission to foster open scientific exchange in all forms across all disciplines. Our 422 diverse and open access journals, including 413 peer-reviewed journals, LAN 9 conference journals, are supported by more than 115,000 academic experts who share our mission, values, and commitment to providing high-quality service for our authors. Today's webinar has been organised by the Journal Cluster of Neurosciences. Neuroscience is a multidisciplinary field dedicated to understanding the complex and intricate workings of the nervous system. This field encompasses the study of the brain, spinal cord, and peripheral nerves, investigating their structure, function, development, and abnormalities. MDPI's Journal Cluster of Neurosciences has a mission to publish recent advances in this field that have profound implications for understanding the nervous system, treating neurological disorders, and overall improving mental health. The following journals are collaborating to offer the scientific community the most up-to-date research and discoveries within the field of neuroscience. Brain Sciences is one of the journals in the cluster and publishes papers on the topic of neuroscience. Neurology International, Brain Sciences, and then Neuroscience is an open access journal of scientific research on neurology and neuroscience. Our aim is to provide an advanced forum for scientists to share their novel ideas and solid research in order to promote the crucial field of neurology and neuroscience. Neurosci publishes comprehensive reviews, original research papers, and high-quality communications, as well as special issues on particular subjects. Neurology International provides an advanced forum for studies relating to all aspects of neurology and neuroscience. Clinical and translational neuroscience focuses on translating or developing basic neuroscience research into novel clinical neuroscience applications and therapies. Neuroglia encourages the publication of timely reviews and commentaries on topics of interest to the neuroglial cell community. Psychiatry International aims to publish innovative studies deriving from original, clinical, and basic research in a variety of fields in order to improve the diagnosis, treatment, and prevention of mental, emotional, or behavioral disorders. Clocks and Sleep publishes original research covering all aspects of biological rhythms and sleep in all organisms. And finally, the Journal of Dementia and Alzheimer's Disease publishes papers that offer insight into all types of dementia with an emphasis on Alzheimer's disease and studies on memory. Today's webinar will run as follows. So we will hear from our chairs who will provide a brief introduction into the topic of today's webinar before we move on to each of the presentations. Each of the presentations will be followed up by a short Q&A, so please make sure to post your questions in the chat box whilst our speakers are going through their topics. At the end of each presentation, our chairs will then read through the questions you have asked the speakers before moving us on to the next speaker. They will then provide some closing remarks before the end of the webinar. If you do have any questions, please make sure to post them in the Q&A box, which can be found at the bottom of your screen. I'll now provide some information about our chairs and speakers before handing over to them for the main body of the webinar. So, Dr. Bognana Drozdowska is one of our chairs for today. She completed her PhD in 2021 at the University of Glasgow, where her research investigated the prognosis of cognitive impairment following stroke. She is currently a postdoctoral associate in the Department of Clinical Neurosciences at the University of Calgary. Dr. Emma Elliott is our second chair and has worked in clinical research since 2014. She completed her PhD at the University of Glasgow as well, focusing on the measurement properties and diagnostic accuracy of cognitive screening instruments. She currently works at the University of Manchester. Ms. Memory Lucy Matambo is our first speaker for today. Her research focuses on post-stroke cognitive impairment in community dwelling survivors and aims to provide evidence to strengthen prevention and health promotion strategies in rural and remote areas of low and middle income countries. Mr. Thomas Aldrieve will be our second speaker. His research looks to combine multimodal biomarkers, including MR imaging, blood tests, genotyping, and demographic data, with machine learning to predict late life cognitive decline in stroke patients. Our next speaker will be Dr. Rodney Ritzel. Dr. Ritzel obtained his PhD in 2016 before pursuing postdoctoral training at the Shock Trauma and Anesthesiology Research Center at the University of Maryland School of Medicine in Baltimore. His translational neuroscience laboratory at the University of Texas Health Science Center is actively investigating the neuroimmune interactions underlying CNS aging, acute brain injury, and chronic neurodegeneration. Dr. Edgar Chan obtained his PhD in cognitive neuroscience at the Queensland Brain Institute and has been working in the Department of Neuropsychology at the National Hospital for Neurology and Neurosurgery, Queen Square, since 2013. He is also an honorary senior researcher with the Stroke Research Group at the Institute of Neurology, UCL. Our final presentation today will be delivered by Dr. Lilian Gao. Dr. Gao's primary research interest lies in utilizing multi-site neuroimaging data to generate more robust generalizable insights for clinical neurosciences challenges, such as conduct disorder and mild traumatic brain injury. She aims to translate her multimodal findings into meaningful clinical applications that can enhance patient outcomes and inform future interventions. I'll now hand over to our chairs to introduce the topic of today's webinar in a bit more depth and to introduce our first speaker.
Thank you so much for joining today's event. We're very pleased to be chairing today's webinar on post-stroke cognitive impairment. Particularly, this is a topic that we feel really connected to through our training and previous research work. As you heard from Meg, Emma and I did our PhDs at the University of Glasgow, actually in the same lab with Professor Terry Quinn. And during that time, we got to do a fair bit of cognitive assessments and questionnaires relating to life after stroke. And as you'd expect, there's a lot of variability in how participants were doing, but still, I remember lots of moments where people seemed just so frustrated or upset and even disappointed in themselves when they realize that they just can't tell you more than 2 words beginning with the letter A, or that they can't recall even 1 word from a list of 5 they just heard a couple of minutes ago. And that really made you appreciate the meaning of these scores that we collect for our studies. It's a reflection of a change in people's lives that affects how they perceive themselves, their self-esteem, and how they feel about their future, worrying that things might get worse and they could develop dementia later on. And it's also a change that can compromise what people value doing most in their daily life. So then we'd go through questionnaires on everyday activities and quality of life. You'd hear how, due to cognitive problems after stroke, people are unable to live independently, go back to their jobs, look after their grandkids, or help them with their homework, or even just enjoy the things they used to most, like reading books. So keeping in mind those shared experience, it's really exciting to find out about all this diverse research going on in this space, which aims to bring us closer to improving person-centered outcomes for those either at risk of living with cognitive impairment following stroke or who are already experiencing it now. So as Meg mentioned earlier, we have 5 talks planned for today with an opportunity to ask questions after each talk. Not sure if there's any other questions that came up or anyone else has anything. Okay, great. Well, that means we were still right on time, pretty much. So once again, thank you so much for that presentation. And our next speaker is Mr. Thomas Aldrieve from University of Calgary in Canada.
All right, I'm going to share my screen here. Let me know if anything's not working or if you guys can't hear me or anything like that. But yeah, thank you for the introduction, and I feel great going after Memory's presentation because it's kind of on the same line, and you'll be able to pick up some common themes. So today I'm going to be presenting a part of my master's thesis on the MRI biomarkers of cognition in transient ischemic attack. I do want to apologize in advance. My voice is a little bit rough. I'm still recovering from a cold from earlier in the week, so bear with me on that. So some background here. Transient ischemic attack, or TIA, is the fancy medical term for a mini stroke. So it is basically a stroke that resolves itself within a few minutes or hours without intervention. And traditionally speaking, these have been seen as fairly inconsequential. In the long term, there's not a lot of impact or long-term changes for the individuals that experience these attacks. But recently, some studies have unveiled that there's actually a very strong connection between these seemingly inconsequential attacks and late life cognitive decline. Sometimes between a 4 to 10 times risk increase if you've had or you've experienced the TIA. So the big question behind my thesis is what are some of these mechanisms that could explain this, right? We know there's this initial vascular event that occurs, and we know that later in life you're at higher risk for cognitive decline, but what connects those two? So one specific mechanism that could be at least part of the connection here is atrophy. So atrophy is just cell death in the brain. We could look at global atrophy of the brain, but also regional atrophy of specific memory-prone regions, things like the hippocampus, or the frontal lobe that are sort of part of those decision-making processes. So for this presentation, I want to go through looking at specifically whole brain and hippocampal atrophy. Again, the hippocampus is sort of in that lower quadrant of our brain. We have a left and right hippocampus, and they're meant to, or it serves a lot of purpose in our memory tasks. So especially short-term memory and converting short-term memory to long-term memory. And in a lot of Alzheimer's disease studies, that's one of the first areas that experiences increased atrophy. That's why we chose that region to look at in this specific study. Our method here is we want to automatically segment these regions. Part of my thesis is I want this to be an accessible tool or an accessible pipeline to be able to use at care centers in the future or a part of research groups in the future. So instead of manually segmenting, as you can see here on the slide, 950 images, it's a couple minutes per image, and it runs a deep learning based algorithm on the image and sort of precisely chooses where the hippocampus and the whole brain is located. The specific algorithm that we used is called FastSurfer. It's been shown to be fairly accurate when it comes to especially volume calculations in brains compared to manual segmentation. I don't have the study on me, but it was out of all the different, I think they tested 10 or 11 different deep learning based segmentation tools. This tool was the most accurate when it came to volume calculations. So for this specific data that we're about to look into, we have just under 500 participants, roughly 60-40% TIA versus healthy control. And then we have imaging at year 0, year 1, year 3, and year 5. So we're going to be looking at longitudinal changes in both the whole brain and the hippocampus in these individuals. So let's step into the radiology room for a second and look at these scans. So on the far left, we have an original slice of a brain scan here. This is what it looks like coming out of the MRI machine. In the middle, we have sort of our pre-processed scan. So we do some corrective factors and some normalization to make sure it fits into our segmentation tool well. And then we pass it into our segmentation tool, which then spits out this final image, which has these kind of red blotches on it. And those red blotches are actually a single slice of the hippocampus. And when I first looked at this, I didn't really like it because they're kind of offset and there's some little pieces here and there that don't look like they're connected. But I wanted to get the full picture to make sure this is at least mostly anatomically correct. So I rendered it in 3D. And this is what it looks like as a 3D model. So we have our gray circle there, that's the whole brain model. And then we have our hippocampus there modeled in the center. And especially when you look at it from the front and from the side, you can kind of see that that's roughly where we would expect it to be anatomically speaking. It's sort of in that lower quadrant. We have that left and right side. There is potentially some improvements there. As you can see, there's some small bits of segmentation that are outside of the main hippocampus there. But overall, this just shows that, at least from a qualitative perspective, this segmentation tool is working roughly how we would like it to. So taking a step back, looking at the data now, first we're going to look at whole brain atrophy. How I modeled this is using a linear mixed effects model that adjusts for age and sex. Like we saw in the previous presentation, both age and sex differences do create a difference in long or late life cognitive decline and would most likely also create a difference in these sort of mechanistic biomarkers. So those are both adjusted for. And what you get is actually you see that TIA patients experience about 6 times the rate of atrophy in the global scale of the brain versus healthy controls, which is definitely a bit more than we were expecting, but is sort of in line with the trend that we expected. You can see about 0.15 volume percent per year for TIAs versus 0.02 volume percent per year for healthy controls. Because everyone experiences some rate of atrophy, the idea here is looking to see if TIAs experience a larger rate of atrophy. And then we have the hippocampal atrophy. This is, again, that linear mixed effects model using age and sex, but also adjusting for whole brain atrophy in this case. So this is looking at how much does the hippocampus atrophy versus the global atrophy that we see in the brain. So is the hippocampus atrophying even more than the whole brain itself? And what we found is yes, the hippocampus is still atrophying at about 2.5 times the rate in TIA patients versus healthy controls. And again, that just fits in line with what we would expect in these sort of in looking at this long-term cognitive decline that we see in a lot of TIA patients. So in conclusion, both the whole brain and the hippocampus atrophy at a much higher rate in TIA patients, about 6 times greater for whole brain and about 2.5 times greater for the hippocampus compared to healthy individuals. And this could potentially provide at least part of the mechanism for this cognitive decline. We know a lot of vascular events are they have a bidirectional relationship with a lot of neurodegenerative diseases like Alzheimer's, and that can sort of explain part of this mechanism here. So the future work, I guess, expanding this into the rest of my thesis, one is correcting for other vascular risk factors. We have things like blood pressure, smoking status, those sorts of things that we could definitely correct for that would increase the accuracy of this model. Correlating with longitudinal cognition data. So right now, we've looked at the cognition data in our specific cohort, and it is declining in the same way that we would expect, where TIAs have a bit more of a sharper decline than healthy controls, but these specific biomarkers have yet to be correlated with that decline. So that would be the next step here. And then also correlating with longitudinal blood plasma biomarkers. That's another big part of my thesis is we have some results for a sub-cohort, and then we're going to expand that out to the rest of the cohort, hopefully, and be able to correlate this all with not just imaging biomarkers, but also peripheral biomarkers as well. And with that, I will, put it back out for questions.
Thank you very much, Thomas. That was really interesting. Thanks for sharing your work with us. So yes, we have 5 minutes for questions. I can't see any questions yet in the chat. Does anybody else have any questions?
I think
Lydia?
Yedian's just raised their hand.
I thank you for the talk. It's very interesting study and data set is invaluable. I was wondering, although you said you have controlled for the sex and age, have you explored there's any sex differences, between like male and females in terms of the atrophy and the cognitive decline?
Definitely. Yeah. So that'll be, I guess, again, part of my later thesis here. I haven't fully looked into all of the sex differences, but similar results to what Memory was saying, where you see females actually having a bit more of experience a bit more atrophy compared to male. But it's not fully conclusive yet. So that'll be part of the final study, but definitely something to watch out for. Yeah.
Yeah, thank you. Oh, can I also ask another follow-up question? Oh, sorry. What is the age range of the sample?
Yeah, so the inclusion criteria for the study is over the age of 45. The average is for the controls was about 64 average age, and then the TIAs were 68 average age. So there's a bit of a range. You know, we have participants in their 80s and we have participants in their 40s, but generally it's in that sort of mid to late 60s on average. Yeah.
Thank you.
Yeah, no worries.
Thank you. Rodney?
Thank you. Thomas, that was a great talk. Particularly interesting, the range that you have a range in patients, the onset of TIA in these patients. Do you notice any trends in the rate of atrophy in those that are younger or older?
That's a great question. I actually haven't looked into that yet, so I'm not sure. I would assume I mean, our bodies sort of naturally, even in healthy aging, we atrophy faster later in life. So I would predict that, you know, those younger participants would potentially have less atrophy compared to some of the older participants. Their brains can potentially offset a bit of that vascular damage. But yeah, I'm not sure. I haven't looked specifically into that yet.
Yeah, it's just very interesting that these are TIAs, right? These aren't actual like
Yeah.
major strokes, and I just think it's very interesting. Do you find that the number of TIAs is positively correlating to atrophy, increased
Yeah,
atrophy?
yeah, that would be another thing that I'd like to control for in the future. I mean, it's hard to tell the number of TIAs that people have had. So the baseline here, or the inclusion criteria, is this is a first-time TIA patient when they come into the study. But obviously, it's 5 years, and these are recurrent events usually. It's hard to track those, unfortunately. So we have some data on that, but there's other times where your patient will have a bit of weakness on their right side or the left side, and then it just goes away, and they're like, oh, whatever, I won't say anything. So there's a chance that that is definitely a major factor here, that some of these patients have recurrent TIAs that can sort of compound that effect. But unfortunately, it's hard to track that sometimes and include that in the data. So we'll try our best, but right now it's a bit up in the air.
Yeah.
Yeah.
That's an interesting comment that Yadin's left in the chat. It would be interesting to know if menstruate affects the atrophy in female participants, if you have that information available. I'm not sure if you collected anything on menstruation.
Yeah. I don't think we did, but that would be an interesting study to do for sure.
Yeah. And did you mention, Thomas, that you have collected data on coexisting diagnoses of Alzheimer's disease and other neurodegenerative?
So again, part of the I guess the inclusion criteria is there's no pre-existing dementia before they enter in.
Yeah.
Now we do cognitive scores across the entire longitudinal timeline. So we do have that data, and that can sort of be used to diagnose patients with something like MCI or sort of some of those early, more general dementia diagnoses. Specifically, we wanted to make sure people coming in didn't already have any sort of pre-existing neurodegenerative disease, at least not at a clinical level, because we want to try and even the playing field coming in. But we do have cognition data across all 5 years, and that's going to be a major part of the project here, ending with looking at how these biomarkers correlate with those cognition numbers.
Thanks, Thomas. There's one question that just came in. Might have just the time to answer this one. Could we look for similar changes using TCD, perhaps in other areas common for TIA?
Yeah, definitely. So I think this is just a very preliminary study on these sorts of biomarkers in TIAs, but we could look elsewhere in the brain. We can look at peripheral biomarkers. I think this is just the beginning. I think this problem of, cognitive decline being at a higher or a higher risk for cognitive decline in TIAs is just sort of coming out now, or at least in the last few years. And there's lots of studies to be done to see what all could be involved here. So yeah, I think that's definitely a possibility.
Excellent. Thank you very much, Thomas.
Thank you so much for that great discussion. And now actually I think it's been great follow on regarding potential mechanisms of post-stroke cognitive impairment. We have Dr. Rodney Ritzel from University of Texas Health Sciences Center.
Okay, if I'm set up here. Yeah, thank you to our esteemed co-chairs here for assembling this group. It's a very important discussion. And I'm going to take a little bit of a sidestep from the path here, but I'm a basic scientist pretty much, and I run a translational research laboratory where we're trying to develop therapeutics. And our more recent interest is trying to develop delayed therapeutics for stroke to improve long-term recovery and prevent dementia-like pathology. So post-stroke cognitive impairment has been found in up to 80% of stroke survivors, post-stroke dementia, about 40%. I think my references are a little outdated, but the numbers are about still the same. So you have a stroke and after a couple years, you develop cognitive dementia impairment. Those intervening processes and mechanisms that lead to that are not really well understood. These would be the secondary injury mechanisms. Um, Over here, you can see that the cognitive trajectories after stroke vary dramatically, but all seem to show some sort of decline. But I'm kind of interested in what the cellular molecular mechanisms are, because there's a big gap in our understanding of what's happening in stroke beyond the first week in the brain, in the immune system. We do know that, like traumatic brain injury, if you look in the brain of an ischemic stroke individual, long after, you'll find evidence of tract degeneration, iron deposition, and actually, among these peripheral biomarkers that Thomas was mentioning, people have found that serum neurofilament light chain in the blood correlates pretty strongly with post-stroke neurodegeneration and long-term outcome. This is all very similar, actually, to traumatic brain injury, which we know is often called chronic TBI because of its enduring neurological consequences. That's kind of where my background as a postdoc was. I studied experimental stroke in my PhD and experimental chronic traumatic brain injury in my postdoc. And I always remember in my PhD, I said, well, when do these stroke mice, you know, people would tell me they just recover after 30 days. I said, wow, they do? I mean, they had this huge like infarct in their brain. And it's like, oh yeah, don't, and the whole field basically doesn't look beyond 30 days in mice. Of course, mouse time is a little different than human time, but even in the traumatic brain injury field, they're looking many months after TBI because if you ask many TBI researchers, they'll say that the mice never really fully recover and actually just sort of decline over time. So when I got this faculty position at UT in a very stroke-rich environment, I thought one of the more interesting things to do would be to ask this question of does ischemic stroke does it really resolve itself after 30 days? It's a super simple question, super simple way to address it. But the vast majority of studies out there looking in preclinical mouse models don't go beyond 30 days. But we know mice can live up to 3 years. So what we did was, for proof of principle, we asked whether a young adult mouse, which is a healthy, pretty resilient mouse, do they exhibit long-term functional recovery, inflammatory resolution, and neural repair 6 months after stroke? So many, many months past where people normally look. And we hypothesized, like TBI, that experimental stroke, which would be the middle cerebral artery occlusion model, induces a chronic neurological disease condition. So we found just a very productive, very amazing graduate student, Rumi Khan, to take up this work. And basically, we just got some young mice. We gave them a 60-minute middle cerebral artery occlusion. After those 60 minutes, the filament is removed and the blood is allowed to reperfuse back in. And we just waited. We pretty much waited, and we measured a behavior at the 2-month time point and again at 6 months. And we just wanted to know, what do they look like? And interestingly, with the proper post-surgery care, we found that we can limit mortality to just within the first week. Surprisingly, we didn't see too much drop off after that. However, they weren't able to kind of maintain their body weight after stroke for those 6 months. When we put them into an open field environment and we just measure their spontaneous locomotor activity, we actually didn't see any changes. So they were able to kind of like scamper about kind of just as well as our sham control mice. But when we gave them a nesting pad and we looked at sort of an activity of daily living, such as nesting behavior, we found that the stroke mice at 2 months and 6 months have a real difficult time tearing up that nestlet, which they would normally do every night. Also, we looked at depressive-like phenotypes. We did a tail suspension test where the mice are hung by their tails, and the amount of time they spend moving is indicative of their despair. And if they spend less time moving, it's indicative of more of a depressive phenotype. And we found that the depression, which many have shown it persists to 30 days, actually just persists for 6 months, maybe indefinitely, but doesn't get worse. I'm not sure if it gets better, but cognitively, we ran them in multiple tests. In a novel object recognition test, the test works such as you show a mouse an object, they become familiar to it, and then you show them a novel object, and a normal mouse will spend more time with the novel object than the familiar object because they're already familiar with the familiar object. But we found that after stroke, many months after stroke, these injured mice have a very they don't really distinguish between the novel object versus the familiar object as well. And actually, it just kind of gets progressively worse. So we do see this evidence of cognitive decline for many years in the field. No one's been able to really show cognitive decline, but they haven't really gone out 6 months. So it looks like maybe you have to actually go out much longer to actually see a decline. And these are in healthy mice. I mean, 6 months after stroke, and these mice are still considered relatively younger, middle-aged. Obviously, Thomas showed you some nice neuroimaging in humans. There's a much cruder method, is we just take the hemisphere out. It's a unilateral stroke injury, so we just take the hemisphere and we can just weigh it, and we see obviously there's a lot of tissue atrophy, which we confirmed using the micro CT. But underlying that tissue atrophy, we took the brain hemispheres and we ran nanostring transcriptomics and in the sham and stroke mice. And what we found was in the MDS plot, so if we look at how similar the brain transcriptomes are between sham and stroke mice, the sham mice brains are here clustered in blue. But the stroke mouse brains at 6 months, you can see there's lots of variation. You have some that you have one mouse here, which we thought was an outlier, but actually there was no rationale no reason to exclude it based on our criteria. It had a stroke, it had all the normal deficits, and all the readouts were fine. It just had presumably a better recovery in terms of its brain transcription. So what did we find? We found hundreds of genes that were upregulated, downregulated after stroke for many, many months after, particularly genes involved in extracellular matrix remodeling and senescence, which kind of makes sense in a way. If you're seeing this accelerated brain aging, then we see it in TBI too, is that you have an accelerated development of senescence as well. Very interested in the neuroimmune mechanisms. When we look in the microglia in the hippocampus, we see that there's a trend for increased immunoreactivity of IBA1. This is consistent with our gene pathway scoring from our transcriptomics, showing a trend for higher activated microglia gene pathways being upregulated after stroke. A little more sensitive technique was flow cytometry, where we're actually able to dissociate the microglia from the brain. And we found that there's nearly twice as many microglia in the brain, and a surprising amount of infiltrating immune cells as well. So just trying to map this onto what might be happening in humans, we work with our local neuropathologist, Dr. Cheryl Tan, who's really helped us kind of identify these postmortem human stroke brains where there's chronic infarct. You can see here, if you look at this area here where the white matter has been hollowed out, this sort of cystic cavity is sort of identifying feature of a chronic stroke, chronic infarct. And if you look microscopically, you'll find that around the border zone, they have a dense rim of activated astrocytes and also activated microglia. So although it seems like this is a very dead zone, it seems to be highly filled with very activated ongoing processes. For our study, though, we wanted to kind of look away from the actual infarct and in an area of the hippocampus in the brain. So Cheryl helped us put together, Cheryl helped us put together age and sex match cohort of brains. These are postmortem brains that chronic infarcts were confirmed by the neuropathology report, and the cause of death was non-neurologic. And we wanted to know whether or not if you had an infarct, remote infarct, if in that ipsilateral hippocampus, was there more signs of neurodegeneration, kind of showing more of a far-reaching effect of stroke on memory. And so what we found was using tunnel stain to label apoptotic cells. We found that in the subiculum of the human hippocampus, there was a significant increase in the number of tunnel positive neurons in the infarct versus our control group. There was a trend in the number of GFAP astrocytes, but a significant increase in the number of microglia in these regions as well, and a pretty dramatic reduction in myelin basic protein, which we use to kind of measure white matter loss. Interestingly, the graduate student, Ramessa, she stained for A-beta 42 immunoreactivity and found that in the human hippocampus, where there was a remote infarct distal, that those subjects had a lot higher diffuse amyloid plaque as well. So if I could just summarize real quick, long-term pathological outcomes can vary widely in younger mice, but it does seem clear that even in young mice, more resilient mice, that they do exhibit post-stroke cognitive impairment and probably even cognitive decline for many, many months after the initial stroke. We see signs of ongoing neurodegeneration, gliosis, inflammation, many months, years, enabling the identification of key secondary injury mechanisms. And this is really important for developing therapeutics to reduce that risk of dementia after stroke. And in a small cohort of human postmortem brains, the remote infarcts were associated with neurodegeneration of the ipsilateral hippocampus. I'm not sure how much time I have, but just want to briefly touch on some of the considerations for these type of studies. You know, the chronic TBI field, the preclinical field has been developing pretty fast since the 80s, but the chronic stroke preclinical field is kind of in its early stages, and so for a lot of stroke researchers out there in the lab that work in this area, there's a lot of considerations such as planned foresight. You know, if you're planning a 6-month study or longer, you're going to want to make sure that your staff is still there in 6 months, or that you're working with the same behavioralist that you consider per diem costs or other grant deadlines. Post-stroke care during that first week improves dramatically. So we've done this in older male and female mice now, and we've taken them out to 6 months to really old age. And actually, if they can just survive that first week, they survive until 18 months of age, which is old age for mice. And lastly, I would just say that the phenotypes are not quite as dramatic in the acute phase. And as these mice get older, they can develop age-related comorbidities that affect outcome trajectories. So these are just some considerations when designing these studies that we came across that might be helpful. But with that, I just want to thank our group here at UT Health. It's a big stroke group, and Dr. Cheryl Tan for our neuropathology help, and our grad student, Burmisa Khan. And with that,
Thank you very much, Rodney. It's a fascinating presentation. It's great to have some preclinical work shared in this webinar. So yeah, we have some time for questions now. We have one in the chat. Thanks for the great presentation. Does the microglio activation co-localize with the white matter lesions? Are they widespread over the brain?
Oh, that's a really good question. We haven't looked at that in mouse or those human, um, but I have seen, I am aware that in the corpus callosum, you will find a pretty, in white matter in general, I think you're going to find a lot more microglial activation. But this is potentially very important, as microglia have been implicated in sort of chewing up myelin or preventing remyelination. This link between microglia and white matter in the chronic phase of stroke is very interesting. I don't know though. Yeah.
Thanks, Rodney. I have a quick question. What are your next steps for this work then?
Oh, I'm sorry if I.
What are your next steps for this work?
Oh, next steps. Yeah, first step actually is so basic, just injure them and wait. Um, and really what we're trying to do is we're trying to gather a bunch of omics data and so that we can really identify these secondary injury mechanisms. What are these processes that are ongoing for years and years after stroke, and we do that through transcriptomics or proteomics. We're looking in the blood. So the follow-up to this is to actually develop a delayed treatment strategy where we don't look at the acute golden window of 3 to 4 hours. We're not trying to extend that to 5, 6, 7, 8 hours. We're actually looking at the delayed therapeutic window that can target um, a month after stroke. It really just it increases that, expands that therapeutic window for most of all stroke survivors. That would really just these type of therapeutics would really just kind of facilitate, improve recovery to the point where they hopefully would not develop dementia.
Really interesting. Edgar?
Hi, thanks, Rodney, for the great talk. I was just interested in the finding with the human data. I'm wondering if you had any hypotheses about the mechanism in which the kind of hippocampal loss, why is the kind of ischemic damage leading to the hippocampal loss? And do you see that in all your kind of human participants, or is it only in like a subset?
Yeah, I would just preface that our human data is based on a carefully selected age, sex match, 5 per group. So I don't want to make too many big leaps of overstatements there, but the mechanism we are trying to, one of the ways that we're trying to assess this in humans. Of course, this is why we study mice, but really we want to know what's happening in humans. So one of the things that we are proposing is we're going to take these brain sections and run a technique called spatial transcriptomics, which actually allows you to visualize the brain section as you would for IHC, but the transcriptomics is done in situ. So you can actually point to a spot on the brain and see what the gene expression transcriptome profile is on that spot in the brain. And we're hoping that that might yield a few more clues. But I'm not sure, that the infarcts were distal from the hippocampus, but there are any number of interpretations. There could just be they were a sick patient or something. There was some other correlation, but we're hoping to increase our N there and try to make a more stronger, conclusive understanding. But that's a great question. Yeah, obviously, we really do want to try to understand the human pathological secondary injury mechanisms too.
Thank
for that
you.
question.
We've still got a couple of minutes if there are any other questions anyone has.
I actually just had a comment to begin with. So I just think that that's super exciting work because, as we know, lots of services are organized just to provide some sort of rehabilitation and support over the first 6 months after stroke, but not much longer after that. So I think having some sort of hope of how you could address longer term issues is, really encouraging to see. And I was just wondering whether maybe you did mention that one mouse who seemed to recover much better than the other ones, which of course, as you said, could be any kind of number of guesses of why that happened. But I was interested whether you'd be looking at the effect of particular APOE genotypes, whether that could affect how people, how mice or people recover after these strokes and how that affects other kind of neurodegenerative changes or inflammation or things like that.
Yeah, that's a fantastic question. In fact, that is something we're kind of interested in for a different project. But in my sort of research on that other project, there's not a lot known about the although ApoE may be a risk factor for sort of dementia and vascular disease. That ApoE4 and long-term stroke outcomes, I don't know how well explored that is, especially from a mechanistic understanding. That's an excellent suggestion and a great risk factor to look at, because it might explain these outcome trajectories, why some people show recovery, why some have a recurrent stroke, why some just decline. Yeah, that's a great suggestion. It's a great idea. Thank you.
Great. Well, I do look forward to seeing more of this work and where it goes in the future. So thank you so much for that great presentation and discussion afterwards. And now back to more of a clinical setting. We have Dr. Edgar Chan from UCLA. Thanks so much. Go ahead.
Okay, thank you for having me. Delighted to present to you the Queen Square Cognitive Assessment Screen. We're going to change brain space completely from mice models to something very, very clinical now. I'm going to talk to you for the next 10 minutes a little bit about this cognitive screen that we've been developing here at the National Hospital in London. So as we've been saying, cognitive changes after stroke are really, really common. They occur in about 80% of individuals after stroke. The kind of complexity or the interest in my perspective, from my neuropsychology perspective, is that cognitive change are really heterogeneous. So you can have very strategic focal infarcts, like for example, a very small infarct in the thalamus can cause very striking memory impairment. And then you've got these very large MCA infarcts that cause like a syndrome of cognitive changes. Changes can be very obvious, like in aphasia and neglect, but then also more subtle and harder to pick up in clinical settings or by family members. And also there's a lot of overlap between stroke events and these other kind of conditions, as we've been speaking about, like vascular dementia, Alzheimer's disease, and so forth, and other vascular pathologies. But if you kind of group and look at the cognitive data in general, what you probably see generally is that in these vascular conditions, including stroke, is a kind of frontal executive syndrome, reduced processing speed, and attentional difficulties. So why is assessing cognitive impairment after stroke important? Well, we know that it reduces quality of life, it increases dependency, and it's good for predicting functional outcomes. It's often reported as an unmet need in the community, often missed by health professionals in appointments and in their interactions with patients. Understanding cognitive impairment is essential to help patients and their families to understand the consequences of a stroke and living with stroke. And it's really important for planning effective rehab strategies. So how do we assess patients' cognitive impairments? Well, in my opinion, ideally, you would be doing a neuropsychology assessment. The strengths of these are that neuropsychological assessments are comprehensive, they're domain specific. Neuropsychologists use normative data. They can take into account things like the patient's age, their education, their cultural background. You can tailor an assessment to the individuals based on what you're seeing in the assessment, based on fatigue levels and so forth. And the administration and interpretation is done by somebody who's had lots of experience doing cognitive testing. Of course, neuropsychologists are not always available. It's a very time-consuming process. It costs a lot of money. So the alternative is to have cognitive screening tools. They're quick to administer. They don't require an expert. Often you can do training very simply and quickly. It's easy to interpret. Most cognitive screening tools have a cutoff score, and you just either impaired or not impaired. It's cheap and cost-effective. But the difficulties and the problems with cognitive screening tools is that they lack normative data. They're not often tailored to the individual. So with most screening tools, you kind of have to complete all of it in order to get a meaningful result. They're often not domain specific, and there's kind of question marks about the validity of the individual items within the screening tool. So can you interpret the individual items of memory or the individual items of perception within a screening tool? So these are a bit dated now, the references, but I think they still stand. A couple of review papers looked at what cognitive screening tools are kind of useful within a stroke population. And essentially reviewing all the kind of screening tools that have been used, up to 30 of them, none of them really fit the criteria that was set out, which is assessing all the relevant domains, being quick, being cheap, and so forth. And kind of what kind of came up about 10 years ago was that the MOCA was probably the best we've got. So in the UK here, the MOCA is kind of the standard screening tool that's kind of mainly used and is recommended by the government, the health bodies, as like the first core screening tool. So how good is the MOCA actually? Well, this man a few years ago passed it with flying colours. So you make of it what you will. I looked at it in this department in this hospital. We compared kind of stroke patients' performance of the MOCA with neuropsychological assessment, and we had 136 patients with subacute strokes. What was interesting was that 30 out of 136 patients passed the MOCA, but actually, if you look at their performance in neuropsychology, 77% of them were actually impaired on neuropsychology, and my criteria for impairment is performing below the 5th percentile, so quite severe impairment on neuropsychology. And 70% of patients were impaired in 2 or more domains, so both kind of quite striking impairment, but also widespread impairment, not just one isolated domain, but multiple domains. And if you kind of plot the kind of percentage of patients impaired in this MOCA intact population, what you find is that patients are most frequently impaired in IQ, in their general intellect, their processing speed, non-verbal memory, and executive functions. And actually, it's these domains that aren't so well tackled by the MOCA, actually. In fact, processing speed isn't measured at all, nor is non-verbal memory. Following on from this, I then looked at a whole other cohort of stroke patients, and what I wanted to see was how well does the MOCA perform in detecting impairment in patients with left hemisphere strokes versus right hemisphere strokes. And the important thing here is that on neuropsychological testing, what I found is the prevalence of cognitive impairment in these two groups are similar. So in the right group, 84% were impaired, and the left group, 92% were impaired. But actually in their MOCA scores, if you look at this line, the MOCA intact score, 29% of right hemisphere patients were classified as intact on the MOCA, whereas only 6% were classified as intact in the left hemisphere group. So my conclusion from this study was that actually those with, that the MOCA is very language heavy, and so patients with left hemisphere stroke who often have aphasia tend to do quite poorly in that. But patients with the right hemisphere strokes that don't have aphasia tend to do quite well, but actually it just then reflects that it has poor sensitivity for detecting any right hemisphere deficits. Of course, since the MOCA and the prevalence of the MOCA 10 years ago, we now have, most of you probably know, the Oxford Cognitive Screening, which has become very popular. It's devised in Oxford. It's stroke specific, it's domain specific, it's aphasia and neglect friendly, and it covers more domains of interest. So there's loads of positives with the Oxford Cognitive Screen, and it's definitely gaining traction and more usage in clinical practice, definitely here in the UK. But it does take a long time to administer, so it takes probably about 20-25 minutes to administer. It's quite resource heavy, so there's quite a lot of pages to flip through. You kind of have to bring a booklet of administration, but there's now an iPad version, so maybe that takes care of that. There aren't really culturally appropriate norms within the UK setting, so they're really only for British people. And the Oxford Cognitive Screen, the original version, doesn't assess non-verbal memory, processing speed, and executive functions. And actually, the team in Oxford have now created what's called the Ox Plus, which do just assess those functions, but that's another 20 minutes on top of the Ox. So that would be quite a long assessment, in my opinion.
Great, thank you so much. I do hope that sometime I'll have a chance to use it for myself. That would be great. Thank
Yeah,
you.
really, yeah. Once it's published, I'll be sure to shout about it. So watch this space.
Great. Yeah, looking forward to that. Thank you so much. And now we have our last speaker. Also joining us from the UK is Dr. Edian Gao from the University of East Anglia.
Good afternoon, everyone. I'm Edian Gao from the University of East Anglia, and today is my great honor to be invited to the webinar. And I'm gonna present the SIGHT trial, a randomized controlled multi-center study testing the spatial inattention grasping therapy for stroke survivors with spatial inattention. And this new therapy is a portable, easy, and low-cost intervention co-designed with the patients to tackle one of the stroke's most disabling effects. With no evidence-based treatments currently available, SIGHT has the potential to improve inattention rehabilitation globally, and this study is going to test the efficacy of the therapy. Spatial inattention is not a vision problem, it's a neurological condition where the brain fails to process information from one side of space. Imagine your eyes work perfectly, but you only notice the right half of your plate, and you start writing mid-page, or sentences appear to begin mid-line on the book, or you dress only half of your body. This is not blindness, it's the brain ignoring half of the world. So after stroke, this hidden disability turns daily tasks like eating, reading, driving, dressing into overwhelming challenges for stroke patients. And spatial inattention affects 1 in 3 stroke survivors. Patients call it frightening, and family reports not enough support as caregiver also struggle. Critically, spatial inattention steals independence away from patients. Compared to other stroke survivors, these patients face longer hospital stays, poorer motor recovery, and lower return to work rate. Over 40% of these patients still struggle 1 year post-stroke. So what has made the challenge even more complicated is that spatial inattention is not just one condition, it's a complex syndrome with distinct subtypes. Every patient's symptom can be slightly different. For example, you will often see two subtypes, egocentric and allocentric, differed by frames of reference. Egocentric inattention means missing objects on the affected side, like ignoring the entire left side of space. On the contrary, allocentric inattention means ignoring half of every object itself, such as only seeing the right side of a cup, a cell phone, or glasses. And it can occur in different spaces. For example, personal space means inattention of your own body. Parapersonal space means inattention within arm's reach. And extra-personal space means inattention beyond reach. Facing this heterogeneity or variations in subtype and spatial domain demands accessible tools that can provide a foundational level of support. And this therapy aims to be exactly that, a core intervention that is adaptable enough to benefit patients despite these subtypes. So how does the SIGHT therapy work? The core of the therapy is a grasp and balance task. To give you an idea, if you have a pen with you, could you please try this with me? First, place the pen in front of you on the desk, and then use your forefinger and thumb to gently grasp it. Lift it up and try to balance it, like this. So you can see it's easy for us because without spatial inattention, we know where exactly to grasp, which is the midpoint of the pen. However, for patients with spatial inattention, this simple task is probably very challenging. They often grasp the pen at one end rather than the midpoint, making it much harder to balance. So our therapy is based on this specific skill to train the patients to put more attention on the neglected space. So instead of a pen, we use a set of 3 rods, which I have one with me today. So you can see they are labeled with different colors and they are in different lengths. So the basic idea is that the longer the rod, the harder it is to balance, which allowed us to adjust the difficulty. We also use a specially designed mat, as you can see here, with colored cubes and letters indicating for each trial where we place the rods. So for each trial, we will give instructions like, please place the green rod on the letter B, and the participant then uses a gentle grip rather than like putting any forces to lift the rod to shoulder height and balance it. And this process is repeated in a structured order throughout a 30-minute session. Here I have a short clip of how the intervention is delivered. Okay, I'm gonna play it.
So I'd like you to use your right forefinger and thumb to lift and balance each of the rods. I'm gonna ask you to place on this mat one at a time. And you're gonna have to lift the rod kind of to shoulder height with your right arm. And then you place the rod back down on the mat on the same position you lifted it up. And you can do as many lifts as you want until you feel that the rod is completely balanced, so like straight. Once you're happy that you've balanced that specific rod, then you tell me and we can move on to the next one. I'm not allowed to tell you how to do it, so you just need to kind of feel in your hand whether the rod feels balanced. There is some temptation sometimes to press really hard, so even though you're not grasping it in the right place, you put too much force and the rod balances. So we'd like you to just grasp it gently with your finger and thumb and lift it up and feel it and look at it and see if it's balanced. Does that sound okay? Well, yeah. So now we'll do the red rod again on letter F. So you put your hand back in the start position, and when you're ready, you reach out and use your forefinger jump to lift and balance the rod. If it feels unrest, you put it back down and again. But if you place the rod back on letter F, try that again, perhaps. Not completely straight as it should be. So let's have another go. Alright, you can return your hand to the beginning if you like. Take your time. Would you like to try one more time, or are you happy with this? Does it feel right? Are you putting a lot of force? It's all how you feel. Okay, go with your hand feeling. It helps you out. How's that? Better? Yeah, perfect. Yeah.
So as you can see from the clip, determining the appropriate grasp point is the key component of the intervention, and the patients must learn to identify it independently, and no verbal or non-verbal cues are given to the patient regarding where to grasp the rod to achieve balance.
So I'd like you.
Oops, sorry. So this therapy uses a key insight. Grasping actions improve attention. So patients use their unaffected hand to balance rods. As you can see from the video, when they pick up the rod, it will immediately tilt if it's not on the midpoint. So if the rod is tilted, the rod provides instant feedback, cueing self-correction. So our pilot data on this therapy from the previous feasibility study funded by the Stroke Association shows significant neck improvement after therapy, with 0% dropout rate in the therapy group versus 37% dropout rate in the sham controls. So we are keen to know what could be the potential underlying neural mechanisms. So previous neuroimaging findings reveal that the intraparietal sulcus, which is a sulcus located on the lateral surface of the parietal lobe, is critical for the object grasping actions. Crucially, IPS encodes how to grasp objects for functional use, and its functional and structural connectivity with the rest of the brain can predict recovery from attention deficits. So therefore, we hypothesize that IPS is a hub for sensory motor integration and is critical for spatial attention recovery. So this side trial has 3 aims. The first aim is to test the efficacy. Does our therapy plus usual care outperform usual care alone? And the second aim is to test the durability. Are the benefits maintained at 12 weeks? So it will be a longitudinal study. And the third aim is to test the mechanism. Is the therapy response tied to both behavioral and neuroimaging factors, including stroke severity, grasping perception dissociation, visual or cognitive deficits? And in terms of the neuroimaging factors, we take into consideration of lesion location, IPS structural and functional connectivity. And for the neuroimaging factors, we'll use multimodal MRI in 80 patients. And for the detailed trial design, I don't want to bore you with those, but we follow the standard order of screening, baseline measures, randomization, intervention. And this page includes the first 2 steps before randomization, and our sample target is 206 patients across 16 UK sites. And then, after the baseline measurement, the patients will be randomized to 2 groups, the intervention group, which will conduct 7 sites therapy sessions plus usual care, and for the control group, we'll do the usual care alone. And this treatment will be done within 10 days. Stratification balances age, neglect, inattention severity, and the parietal lobe lesions. And by balancing this, we can ensure the groups are comparable and can isolate the therapy's true effect. After the intervention, we will measure the efficacy on day 11 and 12 weeks with the most widely used measurements. So finally, we will compare changes in inattention symptoms between the intervention and control groups from baseline to post-intervention. So we hypothesize that when added therapy as usual, 30 minutes per day of sight for 7 days will produce greater improvement in inattention than treatment as usual alone. And the patients with impaired IPS structure or function will benefit less from the therapy. So in terms of the progress, we have 35 sites contacted and 14 sites have received the local information pack. And we have done the SIF for 4 sites. MRI is being set up at 3 sites, and 1 site is open to recruitment. I want to say this trial is a team effort and led by Dr. Stephanie Rosett at UEA. This trial unites the neuroscientists, clinicians, and the stroke survivors and carers who co-designed this therapy. I want to say special thanks to our LEC group, which ensured the patient voices are always heard. We are still open for joining sites. If your group is interested in joining, please do not hesitate to reach out. Thank you.
Thank you so much, Yidian. So interesting. It's great to see a trial being done in this area. We're really excited to see the results when they come out in a few years. We have time for questions, so please, if anyone has any, pop them into the chat.
I was thinking it's probably one of the big challenges is going to be recruitment for this study, I guess. Like always, I guess for most stroke studies, it's hard to get your target participants for research. But it's great you have so many sites on board. That's already very great to see. Sorry, I might have said this, but I missed it. How do you identify the participants? Are they referred from their primary physicians or from doctors in the acute wards?
Yeah, so we have contact with the NHS acute trusts. So when they receive the patients with stroke, they will disseminate our study to them and ask if they are happy to participate. Yeah. So I want to mention like for the recruitment and inclusion, and the patients, they are in the sort of in the acute phase. So from within 1 week to 60 days. So most of them will be referred by acute setting, I would, guess. Yeah.
And
Thank you.
I'm gonna Edgar, go ahead, sorry.
Edgar?
Oh, sorry. Really interesting. Thanks for the talk. It's going to be a great study. I just wondered about the treatment as usual. I mean, it's really tricky, particularly in the acute phase. You know, patients are in a hyperacute unit or acute stroke unit, you know, seeing OTs every day. If they've got fluoride neglect, they're going to be
Yeah.
doing lots of neglect. How much control do you think are you just going to completely leave it up to the teams to decide what they're going to do with the neglect trial? And I guess if I was an OT, maybe I would be like, well, they're in this trial for neglect, well, I'm not going to do my prison adaptation. I'm not going to do my, cancellation task because they're already in this trial. Like, how are you going to manage that? And I guess a follow-up question to that, in your trial phase data, did you find any treatment as usual effects? And was this kind of intervention over and above any treatment, or was the trial data without treatment as usual?
Yeah. So I think for the first question, it's very important. I have raised it myself during the study preparation phase. The short answer is we can't control what treatment as usual each site will give, but we will log that. So during our training for each site, we made it really like clear that please provide what kind of treatment you have provided to the patient. And we will have like the CRF every day. So the either the nurse or the person who is giving the treatment will log that down. And then during the data analysis phase, then we can try to control as much as possible. And that's the best we can do so far because we don't have much control over that. And in the previous study, because the feasibility study, we didn't include the treatment as usual. So we just tested the patients when they are at home. We give them like a mobile watch to measure how many times of therapy they have done. So they are either like doing this or like not doing any treatment. Yeah. Thank you for your question. Yeah.
Okay, are there any other questions?
Thank you so much for that. So that brings us to the end of the webinar. Once again, thank you so much to our speakers for joining us today on our Fridays. So we appreciate that even more. Really interesting research, and I think we all look forward to seeing what the next steps will be with these projects. At the end, I just want to quickly share also just one slide about a special issue from Neurology International, Cognitive Impairment After Stroke. We're accepting submissions now until the 15th of December. So if anyone in the audience is also involved in research work in this area, please have a look. There's the link at the bottom, but I think Meg also posted that in the chat. Great. So if anyone's interested, please have a look, and yes, we'll be really happy to receive your submissions from research in this area. Once again, thank you so much for joining, and to all our speakers. And you have the great rest of your day or evening, depending on where you are.
Thank you.
Thank you very much. Thanks everyone for coming.
Thanks, everyone.
Take
Bye now.